here in this yellow fiber fleece with a model drug curcumin. Additionally, we are also able to immobilize proteins on the fiber surface. Another option is that we can use these resorbable scaffolds for the generation of ATMPs. These are advanced therapeutic medicinal products. And here we were able to tissue engineer skin equivalents and also cartilage on these fibrous structures. We are also able to transfer this material into different shapes. So we are not only able to produce fibers out of silica gel, we are also able to spray dry this material to particles and coat this material by different techniques like dip coating, rotational coatings, or spray coatings. So all these applications I told you in the fibers form is also transferable to particles and to coatings. So we can also generate drug-loaded particles or coatings. And in the moment, we try to go to new applications fields and transfer this clinical tested material into new application areas. And one of them is the cosmetic sector, because our material is really near to nature. Bamboo has its stiffness due to polysilicic acids, which is chemically really similar to our silica gel structure. And a lot of food like rice and potatoes have a really high content of our degradation product, the orthosilicic acid. And this resorption product of our material is known to have benefits on skin formation, the formation of connective tissue, the bone formation, nail stiffness, and hair appearance. And here we can imagine that we can integrate our materials like those particles or fibers into cosmetic products like skincare products, anti-aging products, products for nail care, hair care, and also as a substitute for microplastics because our material is bioresorbable. Instead of drugs, we can integrate others or plant and food extracts into the fibrous structures, and this realizes long-term storage of these others and fruit extracts in cosmetic formulations, and we can also realize the retarded release of these others. We have shown that by fruit extract loaded fibers, and we have proven the bioactivity after this formulation of this fruit extracts. We have particles containing herbal extracts which release these herbal extracts in contact to aqueous solution, and we have odor-loaded particles with a retarded odor release. And the next application field we want to transfer our medical tested and harmless material is the food technology. For instance, for the creation of meat alternatives. And we can imagine that we can use our tasteless material for texturing meat alternatives. We can produce single aligned fibers. We can produce fiber fleeces with a gradient that you have a different mouth feeling in the muscle area of a meat alternative and the more fatty and cartilage area of those meat alternatives. And here on the left side in this green box, I summarized the material properties for the meat alternatives. I think one really important property is that this material is fully tasteless, and we have the possibility of adding flavors to it. Our material is biodegradable to orthosilicic acid. One unique property of our inorganic material is that it's even colorless after roasting or barbecuing. The material is wetted in contact to aqueous solution for an enjoyable mouth feeling, and we are able to adjust the fiber diameters in a range from 100 nanometers to up to 200 micrometers. So we also can guarantee those muscle-like fibrous structures in a diameter range of 30 to 50 micrometers. Additionally, we are also here able to integrate some food ingredients into our material itself. This could be flavors, odors, flavor carriers, color, sourness, or other natural components. And we are not only able to produce fibers out of these materials, we can coat different scaffold structures with our material. In the field of meat alternatives, we can coat food itself, like berries and so on. We can produce particles to be dispersed directly into new foods, and we have different techniques for producing this material directly at our institute. And the last aspect I want to show you today in my presentation, that we can also use this bioresorbable scaffold structures also for the tissue engineering and cell expansion of different cell types. You can see here some fibroblasts and also some adipogenic cells. And I think it's possible to tissue engineer some kind of cultured meat directly on this CE-approved scaffold structure, and we are also able to expand different cell types for meat alternatives. So in the end, I would like to thank you for your kind attention, and I would be really happy if you contact me by mail, or maybe we have some time left for some questions and I can answer your questions. Thank you very much.
Thank you very much, Bastian, Chris. Our next speaker is Dr. Sabine Trump, that will elaborate on sensors.
Yes, good morning. Thank you.
You can share, please, the screen.
Yes, just a second.
Your presentation, please.
Yeah.
Please move to presentation mode.
Yeah.
Thank you.
Okay.
Yes.
Yeah. Okay. Thank you. So, hello everyone. I'm very happy to show our opportunities for collaboration here. So my name is Sabine Trump. I represent the Fraunhofer EMFT in Munich. I'm a chemist. My background is chemistry, but for more than 10 years now, I work in the electrical engineering and microsystems integration with the goal to implement and integrate point-of-care sensor systems into applications. Generally speaking, our institute has a lot of opportunities for manufacturing-oriented microtechnologies. We are silicon technology-oriented in the institute. We have the MEMS line and several clean rooms. I will come later to that point. Also, we have opportunities for fabrication of flexible substrates, so polymer electronics-oriented technologies. I will show you later why it's important to mention that, because we have already a lot of very nice collaborations, also with Israeli partners, also under the frame of the IIA funding. And what we can provide, generally speaking, is to really transfer also ideas you have or we have together really into the product level, and we really can provide continuous manufacturing of technologies and products. So I wanted to show you some sensors. So we have sensors and actuators, and you will see that it somehow even belongs together. Starting with the sensors, as I said, we have opportunities to develop new sensor components. This is very important if you think of, for example, point of care, or everybody speaks about the IoT-ready sensor systems. There have to be sensor components that are able to selectively detect, analyze. They have to deal with the low power and be integrated to multi-sensor arrays. And therefore, we have a lot of opportunities In that case, I show it, it's semiconductor-based opportunities to develop new sensor components. We also combine them with new materials, and this really helps us to enable our sensors to be in various applications. I will show you later on, but first I want to mention the actuators as well. It's very important for several applications to really have opportunities. If you, for example, think of gas sensing, to enable you to improve the performance of your sensors. If you think, for example, of integrated gas sensors in mobile phones, everybody wants to have everything in the mobile phone, or miniaturized devices, for example. You need, for example, some exchange of the air. It's very complicated to wait for the diffusion if you want to have a fast answer what your environmental gas concentration is around. If the sensor itself, for example, is integrated in a small box. That's why it's only one showcase where we can show that we really improve with our microdosing systems. It's silicon or also other materials-based micropumps. We have the smallest micropump in the world, and we develop and integrate the complete microdosing systems to improve the applications. Also, what I mentioned is the polymer electronics-based technologies. So we have different manufacturing lines, I already mentioned. The very important point here is if you think of wearable sensors or large-scale area sensors, you always have to be you have to have miniaturized structures at the end of the day. And what we combine and with our technologies is very important. We have abilities in silicon thinning. That means we can combine the best of the two worlds. We can combine the polymer electronics with already working semiconductor-based sensors. So that, for example, to give you a brief overview about opportunities we can show here, we have abilities to manufacture wearable sensor components and even the whole systems. That's very important to be flexible, as I already mentioned. To show you some use cases, this is some overview about topics we are working on. With components, I already mentioned, we developed whole systems for applications, for example, in air monitoring, so miniaturized low power devices to monitor CO2 and humidity concentrations in one system. Other opportunities, of course, are very interesting if you look for toxic gases or hazardous gases. We develop systems for the soil analyzers to go with electrochemical sensors to measure several analytes and soils. We integrated different kinds of sensors for inline monitoring of water quality. Everybody who works in the water area knows that there are only a few opportunities to go for inline monitoring. And what we, generally speaking, want to provide with our sensor systems is to be able to provide information about analytes in water without sample preparation. So this is really one very important focus to go ahead with technology opportunities. We do water analysis, of course, with environmental interests, but also, for example, you can think of the water analysis in food production and other applications. If we talk about the food, again, if you think of very small and low power sensors that we can bring directly at the point of interest, we have systems where we detect plant diseases in different plants and also really go for the on-site detection, for example, of bacteria in plants. For the food, maybe that's just a nice example where we even can provide systems where we work without any kind of power. So we integrated color changing materials for food monitoring. So in that case, we again use gas sensors to detect the gases that occur during the degradation of food. It's a very, very nice project, and we integrate together with packaging manufacturers, new kinds of materials. But of course, as I said, it's very interesting and important to collect information. So now we go ahead with new technologies where we also combine with electrical readout and data handling, since it will be very interesting for the whole food chain monitoring to learn more about your food all over the whole food chain. With our technologies, I already showed you, polymer electronics based, very, very miniaturized. We are able to even collect data over the whole process. This is very important to learn and improve the quality of food. So talking about the next level of systems integration, this is one European funded project. I just want to show you again, it's flexible electronics based, and the whole system integration is done by us, of course, in collaboration with several partners. In that case, we develop a patch for monitor, so really a body patch where we monitor different information such as temperature, humidity, pH value, and very, very interesting, since it will be it will be used, if it works well, to monitor also the health of the baby, and so it's for pregnant women applications. So we use ultrasound technologies, and this is the challenging part, to combine all of that sensors in one system, to have it flexible as a patch. It should be long-term stable. That means you should be able to wear it at least 10 days. So you have to think of the power source, the complete system integration, and also really to have it very thin and flexible, because it shouldn't be somehow invasive. It has to be a very non-invasive monitoring that we want to provide here. To go to another level, it's also back to the microdosing systems, but also about
Sabine, your time is up. Your time is up. Can you quickly wrap your presentation? We have to move forward.
Yes, it's just to show the last slide, because it's about drug delivery. That's one point, and only to show you that we also really go for the most challenging part, for the implants. We have several microdosing systems where we work with glaucoma therapy and artificial pancreas application, and really integrate the complete system for dosing into bodies. This is what I wanted to say. That's the last slide. Of course, I will share the slides, and I'm happy to hear from you with further questions.
Thank you very much, Sabine. Thank you very much.
Thank you.
We are moving quickly.
Yes. Sorry, I stopped sharing.
Thank you, Sabine. We're moving quickly to Dr. Antoine Dalibard, that will elaborate on efficient drying with superheated steam and added values. Dr. Antoine?
Yeah.
Please share your presentation in presentation mode. Thank you. We are reducing the time to 7 minutes. Please keep up with the timelines.
Okay, thank you very much for the introduction. My name is Antoine Dalibard. I'm working at the Fraunhofer for Interfacial Engineering and Biotechnology in Stuttgart, Germany. And I will present a technology on efficient drying with superheated steam and added value we can get from this technology. First of all, I would like to explain a little bit how the process works. You have to imagine a box which is completely sealed from the top and open from the bottom. It means we are working at atmospheric pressure. What we did in the commissioning phase is to bring the room at a desired temperature, which is normally above 110 degrees, but it could be also higher. We bring thermal energy and we heat the temperature, and then we generate the superheated steam atmosphere, either by injecting spray water or by injecting directly saturated steam. Once we get the atmosphere generated, we can put the material in the atmosphere of superheated steam, and we get like a drying effect because the steam is able to absorb more humidity. And at the bottom part of the chamber, we have a condenser which allows us to absorb the excess steam which is coming from the product, and also recover some volatiles which could be which can have a value, and we can also sell it afterwards. So this part, the condenser part, is very important for us to control, as I said, the steam atmosphere and the layer between the steam and the air. The steam remains at the top of the box because it's lighter than the air, and the air is automatically pushed away from the chamber, and we have a drying process which occurs really in a pure steam atmosphere. Here it's displayed at the batch process, but we can integrate it into a continuous process. This technology has a lot of different advantages. We operate, as I said, at atmospheric pressure, so we don't need complex valves for material charge and discharge. We are free to choose any conveying principle. It could be a belt conveyor, it could be a drum dryer, it could be a vibrating bed. We could also apply this technology for a spray drying technique. Is easy to integrate with further mechanical treatment stage like grinding. We can improve the energy consumption of conventional drying process because steam has a better thermodynamic properties, and we can achieve a better heat transfer. We can recover the heat we supply to the much more easily than with air drying, and if we do so, we are very efficient. Due to the more rapid kinetic, we can reduce the drying time up to 80%, and therefore build compactor system and have a lower operating cost. Another point which is very important also, it's we don't have any oxidative reaction in the chamber because there is no oxygen, and therefore we don't have explosion risk. We don't have emissions, which in many cases for drying process, it's a problem. So it's a completely closed system. The only residues we have from the process is the condensate. And this condensate, we can improve it and taking advantage of it. We can vary the temperature as we like, depending on the application. And the condensate, as I said, might contain, depending on the material we dry, very valuable products. We have applied this kind of technology with many kind of products, minerals, building materials, but also food, fruits, vegetable, nuts, cocoa, coffee beans. We can, as you said, on the top part of the picture here, we can vary the temperature. We can not only do drying between 100 and 200 degrees. We can go for torrefaction and roasting. And we can also, for agriculture purposes, treat a lot of kind of residues, woody residues, sewage sludge, manure, digestate, and especially for this kind of product, we can recover from the condensate ammonium, which can be used as a fertilizer afterwards. So we are very flexible with these technologies. And what we can offer at the Fraunhofer IGB for these technologies, feasibility studies, both theoretical and experimental. We have built different kind of dryers we have at our facility, so we can test depending on the product we receive. We can do scientific evaluation, consulting and investigation. And we have also a team which is able to build some prototype and to design completely a prototype for the needs of the product. And we have also, we are also developing different techniques to separate the condensate and to make useful and available the substances we can get from these drying technologies. We don't have specific patent on the technology, but we can license our know-how and long-term experience. And we can also provide exclusivity for specific application. Thank you very much for your attention.
Thank you very much, Dr. Antoine. The next speaker is Andreas Reimann. He will elaborate on vertical farming. Please, Andreas, share your presentation. Thank you. Yeah, just
thank you very much.
On, presentation mode. Thank you.
Thank you for the nice introduction and the opportunity to speak at this very important online event. I'm Andreas Reimann from the Fraunhofer Institute for Molecular Biology and Applied Ecology in Aachen, Germany. We are very happy actually to present our latest technology development concerning vertical farming platforms called OrbiLoop and OrbiPlant. Actually, we've been looking in the past at different vertical farming and indoor farming applications that are still existing in the market. And we found that several of them are actually working suboptimally. And we wanted to specifically address bottlenecks of the current vertical farming systems. And the outcome of this is OrbiLoop and OrbiPlant. Basically, it is a platform technology based on a very broad process patent, and it allows the cost-efficient and resource-efficient production of food crops, medicinal plants, and ornamentals at a minimal space. And we are using a very new and innovative approach based on a conveyor belt that is going in vertical loops. So we are having full benefit of the vertical space and the high yield, but we are using the vertical production in a much more efficient way. And the good thing also for industry partners is that this technology is ready to be transferred right away. So we have proven the technology for about 2 years internally. There's a broad process patent portfolio, meaning that the industry partner is not bound to a fixed design, but he or she can decide how to develop this technology basis. So there's no limitation in terms of design or flexibility. And actually in Germany, there are already some partners using this technology based on a license, and they are for sure focusing on the German market, but the interest is very big. So what is the concept and how does it work? We have two platform technologies. One, which is here the OrbiLoop concept, is focusing on the in-store vertical farming. So this is a specific market sector where it's not about large-scale production and production of lots amounts of plant biomass, but rather within the supermarkets directly to produce very fresh, hyper-local food, herbs, and also to show this to the customer. So it's a very nice process also in terms of demonstration. So the basis of the technology is a conveyor belt. The conveyor belt is turning around. Inside the conveyor belt, there's a space, and inside this space we are giving aeroponics. So meaning we are spraying a nutrient dust. So by this we already omit handling of large liquids that is usually done with hydroponics or other systems. We have lighting directly here to the sides. So the plants are getting optimal lights. Light recipes can also be established based on the target plant that should be produced. We can easily seed new plants into the system on the conveyor belt, also harvest plants. And this is here not actually bound by any specific location. So this can be freely done. It is very easy to automate the whole system because it's basically a turning conveyor belt and the plants are directly put into the conveyor belts. The roots are growing inside this inner space where the nutrient spray is available for the plants. The plants grow extremely well compared to other systems. So the biomass yields over the time is very high. And here I gave an average of 0.25 kilograms per day. So this is about more than 5 kilograms after 20 days of cultivation per square meter of cultivation surface. And this is still non-optimized. So you can always optimize it based on the light protocol via CO2 supplementation, etc. The system is extremely flexible because you can easily change the height, for example, or the width of the system if you need it. The climate control is also much more easy compared to the classical systems where you have a stacking of the different layers in the tiers, because here basically the generated heat, mainly by the lighting modules, can freely convect towards the ceiling. So this is natural physical convection, and it is easy basically to collect the heat and integrate it into a climate system. The other system that is aiming at the large scale production is the so-called orbiplant concept. And this is here just a representation of our pilot plant that is having 4 different loops. Later on in a full commercial scale facility, you would ideally choose the number of loops according to the cultivation days of your target crops. So the basis is similar. It's also a conveyor belt that is here rotating in this way. Again, we have the inner space where we can bring in our nutrients via the aeroponics. We have the lighting. And here you also see sections without lighting. And this is the reason because you always have a day and night cycle for the cultivation of plants. So there are actually areas where you do not need lighting. And in these areas, of course, you can save the lighting modules, which is also a cost factor. And in classical systems, you always have a full equipment of lighting modules, which is basically quite expensive. What you can also see here is that you have a gradual size of the plants that are growing along this way. So here we have 2 defined interaction points. One where you can bring in the seeds or the seedlings. You put them here directly on the conveyor belt, either as seeds or if you have pre-cultured seedlings. And then the whole system will automatically cultivate the plants. The plants will grow until they arrive at the other end of the system. And there the plants are ready to be harvested. In our pilot system, we do this still manually due to the small size. Yes. But in a commercial, yes?
Yes, we are running out of time. Please wrap up.
Yes, what I wanted to show is also the evolution. So based on the original 1 loop, we have now a very nice in-store farming showcase. Based on the orbiplant system, we have a commercial production concept that is working very nicely. These are just some pictures to show that it's actually working in real life and already there and producing plant material. And also here, for our larger pilot scale system, and you see the different varieties of the crops. You have always a very nice root development. You have medicinal plants, you even have storage plants, but you can also seed at a very high density here so that you have basically like a carpet of herbs or baby lettuce, for example, that is very efficient for the production. Thank you very much for the opportunity to talk. I'll be happy to receive your questions and also later on by email. Thank you.
Thank you very much, Andreas. The next and final speaker will be Professor Stefan Schilberg, who will elaborate on cultured meat. Please share your screen.
Yeah, thank you very much. I will do so. It's loading.
Please move to presentation mode. Thank you.
Yeah, okay. So I will talk on cultured meat and some developments we have performed here at the Fraunhofer IME in Aachen. So just as a wrap up, as Andreas already has told you, we are focusing on commercialization of different products and processes. And I would like just to describe plant stem cells that we have produced for the cosmetic industry. We have developed a non-GMO amylose-free potato that is now growing in the field in Germany for production of industrial starch. We are working together with Continental, the tire company, for production of tires made out of dandelion latex. We have developed a cell-free expression system based on tobacco cells that a company is now using as a screening tool, and Andreas just explains the Orbi plant and Orbi loop technology that is used for vertical farming of plants. I would like to focus on cultured meat and a new bioreactor concept that we have developed here. So this slide is just to illustrate that there are still a lot of challenges in the process. On the left is a process described. I will not go into the details, but just to point out some challenges. For example, to provide inexpensive culture media for cell proliferation, you need scaffolds to grow the muscle cells. You need also bioreactors for meat production. And finally, you have to scale up the process, which is one of the most severe challenges that you have in this process. So we are focusing actually on the whole production chain, also together with some of the Fraunhofer partners, so including other Fraunhofer institutes. But our main expertise is the production of growth factors. We use the cell-free expression system for production of this growth factors, which is meaning that we use a non-GMO process for production, for inexpensive production of these proteins. We optimize the culture medium. The industry is asking for plant-derived scaffolds, so we are also working on that. And finally, we came up with a new bioreactor concept that we also protected by a patent application. And this is shown on my last slide. So the idea is to have a reactor vessel where you have a pistil that is going up and down, and inside you have your scaffold where the muscle cells can differentiate. And by moving the pistil up and down, you bring the culture medium to the cells. And what is most important is that you have a hydrodynamic stimulus towards the cells, which induce the formation of muscle cells, because otherwise you would just proliferate the cells, but not differentiate them to muscle cells. So this special bioreactor concept is used mainly also to differentiate the stem cells to muscle cells. Here you see our prototype. You see that the pistil is moved up and down here. So this is the first small bioreactor that we are using in order to build muscle cells, and you're more than welcome to contact me to discuss how we can work together on this exciting project. Many thanks.
Thank you very much. So we finalized the 5 presentations. We are now moving to questions and answers. If anyone from the Israeli side would like to ask a question, please unmute yourself and we will start answering. Please refer to which technology you would like to refer to. Are there any questions?
If I may, I'm not in really the company, but Sabine, you had a few attempts of commercializing the sensors technology that you have presented. Can you share with us a bit more about what kind of cooperation you are looking for, and what was your experience with this cooperation with Tower you already had?
Yes, of course. I'll be happy to share. So one of several opportunities we always have is, especially if there's some technology input and new developments from technology partners in Israel. So this is how we work, for example, with Tower at the moment. We combine materials development technologies and really integrate our new processing methods into their components. And we will, if you think about the commercial aspect of that, we will deal with royalties or licenses, but we will provide for them the complete manufacturing process. So this is I think, a very nice opportunity, since if there are new ideas that needs to be transferred into prototypes, we can develop in-house the complete process and transfer it to the industry partners.
Okay, thank you very much.
Thank you.
Thank you.
If there are any additional questions that people would like to ask, please.
Please.
Yes, no, I would like, it's Asaf Sagif from Birad, Bar-Ilan University. We are interested in joining into a consortium in the area of food security, water security, and we would like to know what are our opportunities to join such one, if we're not willing to lead it, but just to join it. So of course, if there are any specific company that is interested in participating or for us joining into, we'll be very happy to be connected with.
So we will share all the contact list, so you will be able to contact everyone and see if it's your agenda.
Okay.
Okay, so I will wrap up this session if there are no other questions.
Yes, is there a question, please?
Yes, please.
My name is Ariel. I'm interested about the silica gel fiber. If it's also applicable for span bond or SMS project, not only for needle punch.
Sorry, I didn't get the whole question. Can you please repeat it?
You mentioned that the silica gel fiber is used for needle punch application, right?
It's in biometric application, it's used as a wound fleece for the regeneration of chronic wounds. So you're laying this fiber fleece into wound defect, and the cells grow on this surface structure and regenerate the defect.
Yes, I understand, but okay, fleece is needle punch technology.
Oh, okay.
Yeah, but it's also good for span bond technology?
For bone regeneration.
Span bond, you don't familiar?
No, I'm sorry.
Okay, so, and you coated with a water bone solution or with solvent-based solution?
The production is an ethanolic solvent, and this dry material is directly applied into the wound defect as a dry solid material, and then it gets in contact to the wound exudate and the resorption starts.
Okay, thank you very much.
You're welcome.
Any further questions?
I just wanted to answer. I think it was Asaf. I would be happy if you can contact me, since we will provide some collaboration and search partners for the food security aspects. Since this would be very good if you can send me an email and we go in exchange of information.
Sure, we will. Thank you very much.
Thank you. Thank you.
Okay, so I would like to thank all the 5 speakers of the Fraunhofer Institute. Also the Israeli companies that joined us in this session. We will also share the presentations and the contact of all researchers with the Israeli companies and the academia from Israel. So you can cooperate and see if we can transfer the technology from Fraunhofer Institute to Israeli companies. So thank you everyone for this, for joining us to this in this webinar, and take good care.
Thank you. Bye-bye.
Bye-bye.
Bye.
Thank you. Bye-bye.